article that describes the same thing. Like if I search what is a virus, I might get like a BBC high school level explanation of a virus. If I search what is a virus, PubMed, it pops up like an academic journal that literally does the same thing, but it's now an academic source, right? So it's good practice to start doing these kind of academic sources when you're doing research papers, just because it seems more, it's more professional, and also it's more supportive because it's like peer-reviewed sources. And yeah, and if there's ever any academic journals that you guys don't have access to, feel free to email like the OSIC email address, and then we can unlock the journal for you and send it back to you. So just send us the URL of the journal, send it to us, we can unlock it and we can send it back to you. Does that sound good? Does everyone know the OSIC email? Yeah? Can we can send them like Jamie?
Yep.
We can send them like an email basically with a summary of how to access all that stuff?
Yeah.
Yeah? Sweet. Okay. The next thing, right, so once you come up with your research ideas, the formalities are really, you want to include these three things. You want to have a hypothesis, you want to support it with some form of rationale, and it should be significant in some way, right? The hypothesis is different from a prediction in the sense that it's testable, right? You can measure it somehow. It's falsifiable because it can be wrong. It's like, it's a prediction in the sense that you're trying to see what the outcome of something is, but it's also, it's more of a hypothesis because it's backed by science. You're trying to see an observation and explain the science behind it, right? So like a hypothesis versus prediction, if you take a look at the board, I can break more toothpicks with my hand when it is warm than I can when my hand is cold. Is this a good hypothesis? No, I'm seeing shakes of head. Why not? Okay, what do you need to add?
Like with the addition of something, something.
With the addition of something, something will happen. Okay, interesting. What variables do you need to have in a hypothesis? Very good. An independent and a dependent variable, right? Something you're changing and something you're trying to see the effect of. So in this case, technically it's sort of kind of implied there that we're changing the temperature of our hand and we're measuring it with the number of toothpicks we're breaking. But is there any science behind this, or is this more of an observation? It's more of an observation, right? Like, thank you for noticing. Terrible hypothesis. Next one. This is the classic, like, oh, just add if then. Okay, so if I break toothpicks for 1 minute with my warm hand and then with my cold hand, then I will break more toothpicks with my hand when it is warm. Is this a good hypothesis? No, why not? I was supposed to block out the score so it wouldn't be as obvious, but I realized I didn't do that. But anyways, what's wrong with this hypothesis? It has if then, right? Isn't that the standard definition? What's wrong with this hypothesis? Popcorn style, you don't have to raise your hand, yeah? Okay, sure. Doesn't clearly outline the variables. What else? It's repetitive. Okay, yeah, the English isn't great. Yeah, I agree. What else? First person, true. Yeah, yeah. Anything else? No? Okay, sure. Okay, yeah. In this case, sure. I would argue there's one big thing that is missing. Okay, the condition isn't the same. Sure. That's smart. I didn't even think about that. He was saying that after you break the toothpicks with your warm hand, it might get tired, and then that'll change how it behaves with the cold hand. I didn't think of that. But the big thing that is missing here is an actual scientific concept. What are we trying to explain? Like, yes, like when my hand is warm, I might break more toothpicks. When my hand is cold, I might break less toothpicks. Why? What do you guys think? Why is it that when I have a cold hand, I can't break as many toothpicks? Yeah? Sure, see, that's a scientific explanation. That's not the one I was thinking of. But adding that in will make it a hypothesis. You explain some sort of scientific phenomenon, and then you apply it with some sort of experiment. Something more like this. Well, the English is not great, but we'll see what it says. So if low temperatures suppress muscle contractions and fine motor skills, and I break toothpicks for 1 minute with my warm hand and then for 1 minute with my hand after soaking in ice water for 5 minutes, then I will break more toothpicks with my hand when it is warm. So my beef with this is that it's a run-on sentence, okay? Please don't try to fit everything in one sentence. If you have to break it up, break it up. But what's good about this hypothesis? It has a scientific explanation, right? We've made an observation that we can break more toothpicks with a warm hand. We know that we can break less toothpicks with a cold hand. What is the scientific reasoning? Well, we're saying that if low temperatures suppress muscle contractions and fine motor skills, then we know that our hand won't operate as well. How are we going to test this? If I have a warm hand, I will break more toothpicks. If I have a cold hand, I will break less toothpicks. Right? So in this case, those are the three main things that are added. More examples of hypothesis, right? So most of these are missing a little bit of the scientific background, but again, it has the essential things that you may want to see. Hypotheses don't necessarily need to have the if-then format. It's just usually it's one way to formalize it and one way to like a template, so to speak. But the general formatting of a hypothesis is you have some sort of scientific concept, you have some sort of test to say, if something happens to my independent variable, something will happen to my dependent variable, right? So if skin cancer is related to ultraviolet light, then people with high exposure to UV light will have higher frequency of skin cancer. How does that sound? Right? Do we have some sort of scientific explanation or theory or concept? Right? There should be something before that maybe talks about the relationship between UV and skin. Sorry, were you going to say something? No? But essentially, you will follow this up with your rationale. So if we go all the way back here, right? So you have your hypothesis, you'll have your rationale, and you have your significance. So the rationale is the portion of your research proposal where you're trying to explain, okay, well, why am I supposed to see what I'm going to see? Right? Is there a certain mechanism? Is there certain science? Is there certain studies that have already proven something will work and something won't work, right? How can I apply this to my study to make it a similar mechanism, and why should I see what I see? That's the rationale, basically, right? Significance is just basically like, why is this relevant to society, right? You can say, okay, I figured out a way to break 50 toothpicks in 2 seconds. Congratulations. Is that really helpful, right? And then lastly, you just want to keep it simple. Like at the end of the day, you can come up with the craziest idea. If it doesn't work, it doesn't work, right? Oftentimes, making simple hypotheses, making simple experiments, they'll add up and they'll lead to bigger and better things, right? So you don't need to crane your head and be like, okay, I really, really need this big idea. Sometimes just keep it simple and work from there. So to give you guys a concrete example, I was actually a competitor in the 2016 OSIC competition. That year was Lyme disease. That was our case, right? So if we follow our same procedure as starting with the innovative process, right? They give me all the symptoms. They're like, okay, well, you might have like a target rash, you might have a sudden fevering. There's like this thing called post-treatment Lyme disease syndrome, right? Where you have reoccurring symptoms after you get treated. So I asked myself, I said, okay, what do I really care about? The thing that really jumped out to me is the fact that Lyme disease, it's very difficult to tell you have a Lyme disease infection, and that makes it very difficult for like diagnostics and determining what disease you have, right? So a lot of people get misdiagnosed, and the longer you let the infection accumulate, the more harmful effects can come later, right? So that's what popped out to me. I'm like, okay, why can't doctors figure out that Lyme disease is the infection? What I found in my research, this is me starting to dig, right? I found out that the bacteria that causes Lyme disease doesn't circulate in the body after the initial infection. So it doesn't come back into the bloodstream. That's why it's so hard to trace. Then I thought to myself, okay, is there any way I can turn this around? Because if the bacteria don't move, is there any way that I can attack them and attack the area around it at the same time, right? So then I do more digging. Okay, so what can actually attack bacteria in this way? And what I found was bacteriophages. So they're a type of virus that attacks specifically bacteria. And studies have shown that these are very, very specific. They often can only attack maybe 1 to 2 strains within a subspecies of bacteria, right? Which means that it's a reduced risk for the rest of your cells, reduced risk for the bacteria in your gut, right? Compared to antibiotics, which basically kill everything, right? So this was my idea. And then I did, okay, well, this is a concept. Do I have any proof to back it up? So then I did more research, and they actually do have similar studies of using viruses to treat like salmonella infections, E. coli infections. So it's shown to actually work, not necessarily with Lyme disease, but it has shown that a virus can be used to attack a bacteria. And the advantage of this is that once the virus latches onto the bacteria, it basically takes over the bacteria becomes like a virus factory, and it blows up, releasing a ton of bacteria. And that's perfect because all of your other bacteria are directly around it. Right? So that was my idea, and then that's what I researched, and that's how I came up with my idea. But again, it's the same process, right? So I took this big, broad amount of information. I asked myself a load of questions, and I said, what actually jumps out to me? What do I actually care about? I did some digging around the internet, and I said, okay, well, I figured out why Lyme disease is so difficult to trace. Then I figured out some possible strategies, and then I backed it up with similar studies that had done similar things. And in the end, I'm trying to apply it to my own. So that's kind of like how you back it up with evidence, even though it's something you haven't done yet. Right. So if we relate this all back, so I want to have some good discussion now about this year's case. I was the one of the main authors on the case, and there's 3 cases. So the first one was it was called Conflicted. It was a doctor. He was overprescribing drugs pretty much for everything, and it was taking a toll on him because he feels guilty, right? Like there was this young man that came in, accident, gave him opioids, and he's afraid he's going to get addicted. What are the problems that were presented in this case? Okay, so that's a problem. What is something that you could, like, what can you think about that?
What's their basis for
Very good. Okay, so why do they choose to apply that opposed to another pain medication? What other pain medications are there? Okay, cool. Yeah, there's alternatives. Okay, anything else? Very good though. Thank you, Kira. Yeah. Oh, okay. Yeah. Absolutely. So some people are more prone to addiction than others. Doctors can't tell. Is there any way that you can research something about that? Like, is there a way you can branch off? Absolutely. So what makes people who are more prone to addiction than those people who are not? What makes them different, right? How can you predict that, right? The second case was called Chill, right? So basically it was like a bunch of kids at a party. Kid was getting street drugs, and what he thought was just marijuana was laced with something, and in the end he was overdosing on opioids. What are the problems in this case? You don't have to raise your hand, just popcorn, just shout. Okay, so not knowing what's in there. What can that lead you to? Like what can you, what is something you can build off of that? So I don't know what's in my street drugs when I get my street drugs. What can I do to make sure I know what's in my street drugs? Yeah, no, go ahead. Figure out a way to test street drugs for opioids. Anything else? Right? Anything that comes to your mind, just spit it out. It's like judgment-free environment. Sorry? Produce it yourself. Interesting. Okay. How would you do that? Fair enough. Fair enough. Okay. But yeah, it's definitely starting to think about things outside of the box a little bit, right? Is there anything else of that case that stood out to you guys? I like it. No one knew what to do. Absolutely. So that's very important, right? So what could you do with that? Absolutely. For sure. For sure. So making a drug that is more accessible to kids, more understood, that kind of stuff, right? What is the drug that is used to? No, sorry? Naloxone. Yeah, right? I might be saying it wrong. I don't know. It's one of those chemicals, tomato, tomato. But anyways, that's kind of like the case with that one, right? The third one's called urgency. So you had a paramedic who basically had like the relative who was pregnant. She had, I think, some accident. I don't remember. And then in a lot of pain. And then he rushes her to the hospital and is like, doctor, you need to give her like opioids. And in the end, her baby is born and it's displaying neonatal abstinence syndrome. So now, like Eli talked about a little bit. Was there anything else that was stood out to you?
One thing that stood out to me is like what kind of drugs find that's gonna be as effective as opioids.
Absolutely. So what kind of drugs can we find that can act as quickly as opioids, but maybe without the side effects? What can you branch off from that?
Well, we kind of got into what kind of things that may add to opioids, maybe reduce their harmful effects.
Okay, so what can we do to add to opioids to reduce their harmful effects? What's another important question to ask before that? Okay, so how much opioids do you have to take before the symptoms come? Exactly. What makes opioids have harmful effects, right? So you always want to go back to the cause. The more you can understand, the more you can innovate, right? If you don't understand what's causing it, how can you say that this my new research product or whatever is going to be better than opioids because it's less harm? If I ask you, how do you know it has less harm? If you can't answer what makes opioids harmful, then how can you define it's less harmful? Right? So you want to go back to the basics. You want to analyze all these different aspects of each case. And obviously, go to the one that sticks with you the most and try to go towards it. Do you guys like, I know I asked you at the beginning, but like, have you all decided on the research idea that you want to stick with? Right? That's fantastic. That's fantastic. But I would encourage you now that you have your research idea, again, you want these essential things you want to make sure you cover. Understand what's behind the problem for your specific case. What's causing that specific problem? Try to find some sort of source or academic source that explains that, or some sort of academic source that may suggest a way to improve it, or a similar case that may be used to improve it. If you can back up your idea with evidence from a similar situation, that's the ideal, because you're like, I'll take this technique and apply it to mine, and it should be great. Right? Otherwise, you can't just be like a shot in the dark. Okay, this will work because I think it'll work. It's less strong of an argument, but sometimes if you can explain the theory, it's also equally as strong. If you know how a biological system works and you're saying, okay, well, the interaction between this chemical and this chemical produces this result. So if I change this chemical, I'll produce this result. That's another kind of thing as well. And you just want to provide evidence, you want to provide support, and you want to have a strong argument with your research idea. Does that sound good? Sweet. So I think, how are we doing for time? It's 8 p.m. Do you guys want to take like a 5-minute break, or do you want to just keep going? Get up, stretch your legs, take a drink of water? Or, yeah, 5-minute break? Sure. Go for it. Also, if you have any questions, you can feel free to. I'm not going anywhere. Ladies and gentlemen at home, I've been instructed to give you a summary of what we talked about for the first section of the live stream. Okay, so basically we covered, let me go back to my slides actually. So yeah, we basically covered, so again, whenever you're starting to look at any research idea, or you're reading articles, or you're reading any sort of information presented to you, you want to come with three attitudes, right? You want to be skeptical, you want to be creative, you want to be realistic. In this sense, no matter what information you're given, whether it be in like a Wikipedia page or a scientific blog, or even like an academic journal, always ask yourself questions. So what is this article trying to tell me? What are the key ideas? The information it's presenting to me, is it true? Is it not? What information is used to back that information up, right? You always want evidence and you always want support. And then be creative. So how can you build off of these ideas? Is there anything in these ideas that kind of bother you? Is there anything you'd like to look more into? And then see where you can go with it, right? What can you improve? What can you change? From there, you want to be realistic as well, because some great ideas, they sound great in theory, but they're not actually achievable. You want to make sure that no matter what you come up with, you should have a solid test in mind. And every time you're trying to come up with an innovation, just keep that in the back of your head, like, okay, well, this is something I want to look into. how will I test it, right? How can I research it? What are the variables I'm going to look at? What are the different trials? How am I going to do it in a lab? Am I going to do it with animals? These are the things you want to consider. Next, we watched a video of a woman who went on Dr. Phil and basically had a miracle product that she claimed could do a lot of ridiculous things. The YouTube link will be included in the slides that we send to you guys as like a summary, but I won't talk too much about it now. It's just basically her making ridiculous claims, and then we had some discussion of why her research methods and her ideas were ridiculous. Then we talked about the innovation process. So again, looking at all the information you're presented and trying to narrow it down, taking notes with what you're given, and then asking a ton of questions, similar to what we discussed with at the beginning about the scientific method. You want to talk about, okay, who's affected, why does it work the way it works, what are the different mechanisms, what problems are addressed by the specific information we're given, what's the science behind it, and then really you want to focus in on the things you actually care about. At the end of the day, if you're doing research about something that you can't be bothered less about, there's really no point in doing research about that because it's tiresome, it's tedious, and you really don't want to be there. If you find something that it rings home for you and you know that it's something that you want to look into, it's something interesting, or it's something that is close to your heart, go for it, right? And then that makes the research presentation or the research process a lot more easy to go through with. Then going into your actual research. So starting online isn't always a bad thing, right? Because you know that looking up Wikipedia or looking up like quick Google searches, they can turn out a lot of useful information very, very quickly, and often in shortened summaries that are easier to understand and capture more information in a smaller amount of text. However, no matter what you find online, you always want to find an academic source for it to back it up. So you may do a quick Google search and someone wrote on their blog that a certain virus behaves a certain way. Great. If you can do the same Google search, and like one pro tip is just replicate your Google search and then type in PubMed after, it'll usually pop up an academic journal that talks about the exact same thing, but it's an academic journal, right? So it's a peer-reviewed source, and you know that it's, for the most part, reliable. If you're looking into some information and then you do an academic journal search and you find that the identical information is nowhere to be found, could that suggest something? Maybe that the information is not necessarily backed up by science. That's for something for you to decide. In terms of journals and how to access them, if you ever find a locked article, you can always send the URL to the OSIC email. We will send you like a summary email of how all that's done. But yeah, so feel free, don't be deterred if you see an article that is locked. We can definitely access those for you. And I think I covered the rest of this. Yeah, okay, sweet.
Let me check us. All right.
Yeah.
Yeah, that was
I'll be in the background.
all right.
The ideas. Ambitious.
Okay, we're gonna continue now. We're gonna. I don't know. Is it? Did you? Can, all righty. Can you guys hear me? Good. Everything's good. Okay. Perfect. So for the methodology section is basically going to be the section in your research proposal that will elaborate the steps and the experimental techniques that you're going to use in order to either affirm or to refute if your hypothesis is correct or not. So basically, for your methodology, okay, so the details for your methodology, the methodology is not, like some of you may know, it's not your basic materials and methods list that you have in your high school lab report. So it doesn't follow step by step. It's actually going to be a bigger text. So you're going to have each step you're going to use for your protocol, but you want to add an explanation to it as to why are you using that step. What's the purpose of this step in your methodology? What will it allow you to do? Like what conclusions will it allow you to come to? Your experimental details are not needed, so you don't need exact volumes or specific steps. So for example, if you're using a solution of NaOH to catalyze a reaction, for example, you don't want to include the specific volume and the specific concentration of NaOH. You just want to say, for example, I'm going to use NaOH as a catalyst for this reaction, which will allow me to get this, for example. You also don't want to include specific steps. So for example, if you're using, let's say, gas chromatography mass spec, which is a method to quantify the amount of a specific chemical in a substance, in a solution, you don't want to say each specific step that you're going to use for this technique. You just want to keep it general. So you're going to say, we're going to use gas chromatography mass spec in order to quantify the levels of X compound in our solution. So don't include those specifics for each step. You want to make sure to mention your independent and dependent variable. So if some of you may not know which one's which or what your independent and dependent variables are, your independent variable is a variable that you control, that you will modify in order to see the behavior of the dependent variable, how your dependent variable will change. So your independent variable is the one you control and will allow you to see the change on your dependent variable. You want to make sure to have a reasonable sample size. So in a lab, you can't just have as large of a sample size as you want in order to have the most viable data. Because if you have a larger sample size, that comes with a cost issue. So you can't have such a large sample size. Then again, you can't have a sample size that's too small. Because if you have a sample size that's too small, you don't have enough results that will allow you to make a good conclusion. So in some cases, when you have too little of a sample size, you end up making biased conclusions. So in accordance to your conditions, to what you think is essential for your research, for your methodology, you want to pick the most, the best sample size for your methodology. Okay, so for in a lab, we have in vivo and in vitro settings. So in vitro is basically when you're doing anything that has to do with working with cell culture or tissue culture. So in vitro is the lab techniques that you will be doing at the cellular level. You could also have an in vivo model where you're working with the animal itself. So that being, it could be a mice, it could be, mice, it could be Drosophila, it could be any animal that's used in a lab. So you want to, if you want to choose one of these models, and so in accordance to the definition of each, you want to choose the proper one and see which one, why it's the best one according to your research. Okay, so the specifics of your methodology. I'm just gonna have a sip of water. If you guys have any questions at any time, stop me and I'll be more than willing to answer them.
So we have to stay specific, in vivo or in vitro?
In vivo and vitro. Yeah, so you want to stick to one of them.
Just one.
Yeah, stick to one of them. You don't go from one to the other. That's right. Oh, you did both?
Because I talked about like you do in vivo or in vitro first
Okay.
before you do in vivo because it's not.
Okay, I see. So do you want to share?
Okay, I mean, like for me, Eli, to answer your question, the year I did my study, I did in vitro first and then in vivo. And I think most times the research process is more catered that way because if you start in vitro, you're doing it in like petri dishes, you're doing it in a lab setting without it. It's more of an ethical step to do a proof of concept with like cell dishes or whatever before you put it in living organisms. Like, okay, I hope this works. Like it's just better to do it in a more theoretical setting and then move on to living organisms. But if that's assuming it applies to living organisms. If it doesn't apply to living organisms, then maybe just Good question.
You said like a reasonable sample size. I
Yes.
was just wondering, like, you get to choose your own sample size, right? Like,
Yes, exactly. So you choose your sample size. Yeah.
you just don't want to do something that's too much that wouldn't be able to like be possible in real life. Is that what you're saying?
Like,
Exactly. So what I said, you don't want to pick too large of a sample size because in reality, you can't afford such a large sample size. It would be too costly, right? So for example, if you say, oh, I want to use a sample size of 100 mice, well, doing your test on 100 mice is quite a bit. So that's why you have to find the middle in between a sample size. You don't want a sample size that's too small because you won't have enough data, or you don't want it too big because then it'll be too costly and it'll be too large of a project to run.
there's a lot of gray room with the number. Like, there's no perfect.
Yeah,
number.
exactly.
It's just as long as the number seems to sound about right,
Mhm.
go with it, right? You don't have to be like, okay, like, I need exactly.
Yeah,
Does that
exactly.
make sense? How can I, like, it doesn't matter specifics. As long as it's not too small, not too big, sounds about right, go with it.
Any more questions? Okay. So we're going to continue with the specifics of the methodology. So you want to make sure to address all the components mentioned in your hypothesis. So as George mentioned, your hypothesis is kind of like an educated guess to what you anticipate for your proposal, for what your research is going to give you. So you want to touch every aspect of your hypothesis. You want to basically, through your methodology, you want to come up with results that will allow you to answer your hypothesis. You also want to try to clearly outline the manipulated and controlled variables and why you're using them. So that being, we'll get to that. We'll get to that here. Actually, we'll go through it now. So in an experiment, you're going to have two variables. You're going to have your control group and your experimental group. So your experimental group is the group, your sample, which you will be administering your test, administering your procedure. What you're trying to do, you're going to do it on your experimental group. Whereas your control group, it's the group where you won't be applying your procedure, where that's the group where you won't be administering any tests. So the purpose of having these two groups is to see the variance in between so what the results that the experimental group will give you will allow you to see how it varies from the control group. So the control group will act as a baseline, and you can see how the results for the experimental group vary in function to the control group. You guys get that? Go for it.
To the, does it control group and the have to be,
Okay, so yeah, I could talk to that. So basically, you want your control group and your experimental group to be submitted to the same conditions to eliminate any confounding variables. So that being, you want them to feed them with the same meals. You want the mice to be approximately the same age, to be in the same like cage kind of thing, submitted to the same environment. The only difference between the two groups is one you're doing your test on, and the other you're not.
How will they say that in the methodology? Should they go into all the details of what they have the same conditions, or should you just do one line being like they're raised in the same environment and the only thing that like with all other variables controlled, we
Yeah.
only alter X.
Okay.
Because like I'm just asking, should they in the methodology, should they say if we raised them with this condition, we gave them that food, we gave them like,
Yeah.
right? Should we say all that? Or should we
Okay,
say-
so what I would suggest for that, you could keep one brief sentence as to what the conditions are, and you want to specify that you're going to have them, you're going to maintain them for both groups. You want to have those conditions universal for both groups. And you want to say, you also want to add a brief sentence at the end of your methodology, what you expect in your experimental group as to your control group where you don't expect anything to occur. Does that answer your question?
Yeah, no, I was just saying because I believe there's a word.
Yeah, that's right.
So you just don't want to be like, okay, we raised them with food,
Yeah,
and these conditions, and we gave them this amount of sunlight, and they had this many friends. You don't need to go into that, right? Just be like, with all of the variables controlled, we changed this variable. This is what we investigated. In our experimental group, we expected to see this. In our control group, we expect to see this. If there's a difference,
Exactly. Yeah. Any questions about the experimental group, control group? Nope. Okay, good. All right, I could also go into some detail about the control group. So in some experiments, you do subsection your control group into two controls. There's a positive control where you expect a result where you expect your result to happen, and your negative control where you don't expect any result to happen. So for example, I'll put a context to this. So if you want to test the degradation of a certain compound, a certain molecule, whatever it is, in a solution, your experimental group would be that solution itself, and you're going to test different techniques of degradation on that solution to see if your X compound, the compound that you're analyzing, is being degraded. Your positive control, where you do expect a reaction to occur, a degradation to occur, is a pure solution of only your X compound. So it won't be the whole solution itself, like the reaction mixture. It'll be only your X compound. So in your positive control, you expect that degradation to occur because it's only that compound that you have. For your negative control, it's going to be your reaction mixture minus your X compound. So in that case, you don't expect any degradation to occur. Does that make sense?
Want to give an example?
An
I'm a
example
little
to that?
confused.
Sure. I don't know if I should get a visual aid for that. Okay. We didn't use positive and negative control for that.
Good practice. Good practice. Also, this is what you want your methodology to look like on the final poster.
On the
Obviously, in
poster.
the proposal, it should be like list of steps, but for the poster, please do this. I made a mistake. I ran out of time, okay? Procrastination. But I basically just put a bullet in
Oops.
this thing, said like, how much things? Make it a flow chart, make it nice and beautiful. It should be self-explanatory.
Yeah, so that's for the final round, not what you guys are doing right now, yeah. Do you guys want an example for that positive negative control? Okay. I could think of one. So for example, if a group of researchers are trying to make a treatment for a specific cancer, and you have your cocktail, which will act as treatment for that cancer. So you want to see if that cocktail actually works as a cancer treatment. So for your experimental group, for that study itself, your experimental group will be the whole cocktail. So the different things that you'll be using in that cocktail, such as the active ingredient, the different fillers, things that allow the active ingredient to work, or a buffer solution, other things like that. Everything that's in your cocktail, that will be your experimental group. So the cocktail itself, administered to the cancer patients. Your negative control group will be, you guys can correct me if I'm wrong about this, but your negative control, so if it's where you don't expect a reaction, a response, it could be, I would say it could be where you're doing your treatment on, what would you guys say to this?
Yeah, so like, sorry.
Yeah, go for it.
Essentially, so the negative control, you will know that this should not affect the cancer whatsoever.
Mhm.
That's why we call it a negative control. So something that you know will not do anything. Give them water. Right, right. But
Okay.
the purpose is you want to make it as close to your experimental drug as possible,
Mhm.
minus what you're actually trying to test. So for example, let's say the drug is drug X, and then when you're administering the drug, you have a syringe, you have saline in there, you have maybe some stabilizer, and then you have your drug X. So your negative control, you're going to only put everything but drug X. So you'll still inject people, you'll still include the saline, you'll still include the stabilizers, but you will not include that drug X that you're experimenting with. And you know that those other ingredients are not supposed to induce any change in the cancer. So then you'll use that as your negative control. And then your positive control is something that you know will have an effect on the cancer. So that's, for example, like a known chemotherapy drug that you know will affect this cancer. So you will use that to say, I will compare my drug to this known chemotherapy drug that's known to treat the cancer. And I know that this will have an effect on the cancer, so that's my positive control. So you have something that you know will do nothing, which you want to make it as similar as possible to your experimental drug. You know something that will work, so that's a positive control. Then you have your experimental group, which is what you're hypothesizing is going to work in your case. Does that make it clear?
Thank you. All righty. You have any questions regarding that?
Do you have a group that you do nothing to? Like, that you just go through?
No. So the experimental group is going to be like you will have a group where you're not going to administer your experimental whatever you're testing, right? But you want to make it as close as possible to that group without actually doing your treatment. Because okay, so let's make it like more concrete so that you could understand. So let's say I'll use the example of plants. Let's say you're testing some sort of medication to make your plant grow, right? So you're gonna have like different groups. You're gonna say, in a subset of plants, I'm gonna put the medication in water and I'm going to water these plants with the medication. And the other group, I'm gonna have plants and I'm just gonna not give them the medication and see if they grow.
So water.
But you would still put them in the same environment. You would still give them water the same way, the same volume, the same amount that you would in your experimental group. So they're very close, but the only thing that's missing is the drug itself. And that's to make it comparable, because otherwise, someone could say, I will put these plants, I'll put them in a different environment, like no light, no water. And then this group, I have the water, I have the light, I have my experimental drug. And then someone could come to you and say, wait, they had different light exposure, they had different watering treatments. So how can you say that's your drug that's doing the trick, right? So that's the purpose of splitting your groups up, but not too different so that someone can't come to you and say, well, your results can be different because of this reason and not because what you're trying to tell us. Yeah.
So if I'm understanding right, you have your control group has 3 different control groups. So like one where you have the negative, one you have the positive, one you just have environmental setting.
Well, you'd have 2 controls and 1 experimental, right? You have a treatment group that you're looking at, and then like, this is what I should see if it doesn't receive the treatment. This is what I should receive if it has something that's proven to show me something. And then this is my experimental group. This is the new thing I'm trying. Is it closer to nothing happened, or did it work? And it's closer to something that we know will have some sort of outcome.
Okay, so the control group only has the positive and negative.
Yes. Technically, they're 2 separate control groups,
Yeah.
right? A positive control and a negative control. And then you have your treatment. So ideally, your study should have 3 groups, 2 controls and 1 treatment. Yep.
You know,
What do you think? Sorry? Assuming equally, why would it be equal?
just because I would have, you know, the sample on
Sure. Right. So like, again, sorry, I'm not trying to, I was pushing on the spot a little bit, but it's like, absolutely, you're correct. Like, that's what you would like to, you want to keep most of your variables as similar as possible. If you have multiple, if you have like 3 in one group, 3 in another, 3 in another group, does it make sense to have more in one group than another group? Not necessarily, right? Because you want the variability to be roughly the same, if there is any variability. So absolutely, I'm sorry. Put you on the spot a little bit. You're absolutely right. Same amount of, samples in each group, same conditions, whatever you're changing, that's what you want to see with the positive. Yeah. Mhm. Sure. So like we know that this fertilizer has been in a study, and we've seen that if we add this fertilizer, the plant will grow 25%. So we know like there's something behind it that we know it will cause a certain outcome. Does that make sense? Because it's not like we're trying something else that we don't know either, and we're like, maybe it'll grow with something that you should know and all understand. That makes sense? Any other questions?
No.
Okay, Justin?
All right. So to go back to the specifics of the methodology, you want to try to clearly outline your manipulated variables. So that was the groups that we just spoke about, your control groups, that being your positive and your negative, and your experimental group. You want to also mention any assumptions that you're making. So for example, in your research proposal, if there's not enough information on a specific topic from prior research, then in some situations you're going to have to make assumptions. So for example, if I'll take the example of, and this is what the methodology here is about, CRISPR. How many of you know what CRISPR is? Okay. So CRISPR, for those who don't know, it's a new gene editing tool that's widely used in labs nowadays, and it allows a specific knockout or any type of gene editing in any organismal genome. So basically, I'll get to that now actually. So on the same case topic as George participated, I myself participated with someone else. And for our methodology, we proposed that, well, I'll give context to it first. So how we came up with our research idea, we looked at the different factors that came into play in the transmission process, the transmission process. So when a tick is feeding on a host, we looked at what happens during the feeding process. So the bacteria is being transferred from the tick, the Lyme disease bacteria is transmitted from the tick to the human, and there's other things that are going on. And amongst those other things, we noticed that there was this protein called SLP15. You guys see that in the first section of the methodology, which was a protein that was immunosuppressive and inhibited the activity of some specific killer T cells from the human immune system. So what we decided, we said, okay, since when ticks feed on humans or any host, they release this protein that is found in their saliva into the host that inhibits the human immune system. So we said, okay, how can we remove this protein so that there will be immune response, so that the immune system will target the Lyme disease bacteria and eliminate it. So for our methodology, we said, okay, we're going to look for a gene editing tool that can knock out the gene in the tick's genome that codes for the production of the SLP15 protein. So through the series of our methodology, we'll have our two groups. Our control group, we didn't use a positive and negative control, but we have our control group and our experimental group. So the experimental group is where we'll microinject the ticks with the CRISPR complex containing the specific RNA sequence that matches the DNA sequence that codes for the SLP15. So once we microinject those ticks, we expect there to be knockout for that specific gene sequence, and so that the offspring of those ticks will not produce the SLP15 protein. So that was what our methodology is. And then what we do is we allow the offspring of the gene-edited ticks to feed on mice. And this model is obviously an in vivo model. So we allow those ticks to feed on mice, and then we analyze the tissue from the bite area to see if there are still levels of the Lyme disease bacteria in the ticks that we edited and we removed the SLP coding gene. And then we would compare both groups to see if there was an effect, if there was a difference between the two. So that was our methodology. Do you guys have any questions regarding that? Okay.
Also, just something to think about. So back when I did my methodology, so again, I was working with viruses and bacteria, and one of the questions that came out to me, so it wasn't in my methodology, but one of the questions I was asked were, will there be any side effects to you adding these viruses into the bud, right? My answer is kind of dumb. I was like, well, I don't know. Like, gotta find out, right? I was saying in my theory, I was like, well, they're very specific, so they only attack very specific bacteria in your body. That way, What could I have done to test that? Like in that presentation, what could I have said to the judges? Any ideas? How can we test the effect of these viruses in the body without actually being in the body? Sample cells, absolutely, right? If you're in a living system and you're afraid of the impacts of the living system, what would these viruses interact with? Assuming it would be some sort of immune response, maybe some of your immune cells would attack the virus, right? If they do that, is there some sort of response? Would you get fever? Would you get side effects, right? To monitor that, you take immune cells from the body, right? Put them in a petri dish, grow them, and then see if you add the virus, is there some sort of. So you wouldn't include this in your methodology. This is just something extra to think about for when you're actually in your presentation. If people ask these kind of questions, and these are questions you can ask yourself as well, you prepare to answer, right? Instead of the, I don't know, right? You're like, we could look into it with this other experiment, with this other setup, and actually still try to look at it. Okay?
Any more questions? Nope. All right. Okay, so you want to make, so to get back to this point, mention any assumptions that you're making. So if we go in the context of the methodology that we took, let's say, for example, there's not enough research, previous research that was done as to which which virus to use to infect the tick cell in order to introduce the CRISPR into the cells of the tick. So how CRISPR works is you need to insert the CRISPR-Cas9 complex into a virus in order to infect the cells of the ticks and in order for the edit to occur in all the cells. So, for example, if there wasn't enough research done as to which virus to use in order to infect the tick, then we have to make an assumption. We look at the different factors, the different conditions, and see, we make a decision saying, okay, we'll use, for example, the HIV virus to infect to infect the ticks, and for the CRISPR-Cas9 complex to be present in all the cells, and for the edit to undergo. So you want to mention those assumptions that you make. You also want to address any potentially confounding variables and how you minimize their influence. So that's the purpose of maintaining those same conditions within your control groups and your experimental groups. You want to, as we previously spoke, you want to keep the conditions the same. So that being the food, the cage, everything that you're doing to the mice. Okay. Do you guys have any questions regarding that? No? All my slides. Yeah. All right, so do you guys, I think that concludes our presentation. Do you guys have any questions?
Overall,
Anything?
Not really about the presentation, but I was wondering if I can cite work that cites other works.
Yeah.
That's fine.
Usually you cite the original author when you're, so let's say the article you're using says this person showed this. Usually you don't cite the reviewer, you cite like the original work. So if you could find where that information originally came from, do it. If not, just feel free to get it from where you took it from. Yeah. That's just extra work, I think.
That's extra work.
Yeah, it is.
Yeah.
Good on you.
You can submit it in any language you want. I would just stick to one, honestly.
You're
Yeah.
just looking
Oh, and you can't. Oh, interesting. Okay, I would say for the written phase, I think it would be best that you can't submit a written proposal like half one language, half not. So for the written one, just submit one language, doesn't matter which one. For the oral presentation, then you guys could do half. But your poster will end up being in one language. You cannot print two posters in two separate languages. But if you're really comfortable in one language, we'll have bilingual judges so that you could present in the language that you want. Yeah.
Sure.
Interesting question.
Yeah, that's a good question.
We could run through a small like exercise so that you guys could like put it into practice what we discussed. So we could do one right now. It'll take like maybe 6 to 7 minutes, and then we'll see if you guys understood the presentation. The stream is still going, so they'll be able to. Oh, that's fine. You can just leave it there, though. Yeah, sure.
I apologize for this. I got it.
Yeah.
Other competitors of the competition.
Okay, so someone give me a random topic that we're gonna. So this is to practice the methodology, how to make a methodology. Someone give me a random topic, like something that we're gonna test, like something random. What is it? Jilly juice.
The lady at the beginning who says drink rotten kale and it regrows arms.
Okay, sure. So we're gonna use that for what? What are we testing? Is it regrowing arms?
I don't.
Yeah, basically.
Regrowing arms. Okay, that's a good example. Okay, let's go with that. All right. So let's work with that. So she's hypothesizing that this juice is gonna regrow arms, right?
Because
And-
the integumentary system cut it will heal, so you can heal your arm. That's the science.
Right, so that's actually a really good example to use right now because it's a little complex, so it'll be good. So an arm is composed of what? What is an arm composed of? Bones, muscles, nerve, vessels. Right. So if we're gonna test if the juice works, we would have to see if this juice could regenerate all those different structures, right? So obviously it's gonna be hard to write a methodology on every single one. So a good scientist would probably focus on one aspect that she wants to test. Is it gonna regenerate skin, for example? So we're just gonna use that, okay? So we're saying that we have this juice here. I'm gonna draw the juice. And this is
It's green.
that's green, okay, so I'll just use green. I think the people on the live stream are gonna be very confused as to what's going on. But they could watch the YouTube link and I guess they'll understand.
The first like 6 minutes.
Okay, and I think it was kale. I didn't actually watch the video, but it's kale.
It's like kale and salt, yeah.
Okay, so we're gonna say kale here. Okay. So then we're gonna test this, and we're saying that this is gonna be good for skin regeneration, right? Okay, great. So the first step is to see what is skin composed of, right? It's composed of epithelial cells, so EC. I'll just abbreviate it, so I don't want to type it every time. So epithelial cells, okay? So to test, so I think the first step is to see, okay, how can I test if epithelial cells are able to regenerate themselves? So I would look up online and see, like, is there any way we could work in a lab and see if epithelial cells can regenerate? And I'm sure nobody here is going to know what. But there is one test that you could do, which is you could have a petri dish and put a bunch of cells on there. Okay, so this petri dish is full of cells, and then you could actually scratch it. This is actually real if you look it up. You could scratch the petri dish, and it leaves a part of the petri dish that's blank. And then you could wait a couple days, and then these cells will slowly reproduce and then fill that gap. Then, depending on how fast that happens, or if it happens at all, you could know if these epithelial cells have regenerated. Okay, so we know that we want to test the active ingredient in this, what is it called? Jilly juice? Jilly juice, which is kale, okay?
And salt.
And salt, okay. So what do you guys think so now knowing that we have to do this petri dish cell scratch test, how would you make an experiment to test this? Like, what would you do?
You would do a first experiment and then that adds like your control, I guess, and then do the same thing, but add the kale piece and
Okay.
see the time, like, we'll do the same time.
Yeah.
Okay, so would the control be a negative or positive control? One where you
A negative.
Good. Okay, so she's saying just put one that has everything in this juice minus the kale, whatever active ingredient is in kale. So we'll have, I'll just draw quickly the petri dishes. And then you're saying this one is juice, so like all the salts in the juice minus the kale. Okay. And then what would be, so that you're saying that's my negative control because you don't expect any of that stuff to actually do anything, right? Okay, so what's your experimental group?
So,
Or anyone in the room, I don't need a.
It would probably be the cells plus all of the body.
Right, so then your positive control, your experimental group, what am I saying? Experimental group is all the active ingredients in the juice, including the kale, right? So anything that she's claiming that's in that chili juice, that would be your experimental group. And then what would be a positive control? So we know that usually that juice is not supposed to do anything. So what would be a positive control that we could use as a comparator? Yeah. Sure. So you're going to use some sort of drug that you know makes your skin repair faster. I can't think of one. No, I can't think of one on the fly here, but you will use something that you know will make your skin repair faster. And then you say, okay, so that's going to make these cells reproduce faster. And that way I can compare my drug to see if these are the same. And hopefully your negative control and your experimental group are not the same. That would mean that this group right here is actually doing something. Okay, so so you know your groups now. What are the variables that you're going to measure? So how would you say, like, what are you going to measure exactly as in terms of your results to know that one group is performing better than another? Yeah. Good. So you're saying the rate of, I'm assuming you mean like rate of cell division. Good. So that's perfect. Yeah. So then you could basically say also like time, because our experiment is basically to leave a scratch and wait for these cells to replace it. So then you can actually measure the time and see how long does it take for that to happen. That's good. Is there anything else that you can measure? Yeah, so we could talk about sample size, but would there be anything in your results that would help you also? Yeah.
Well, we can compare a normal skin cell to the actual cell that's being replicated multiple times, because it's also dangerous, right?
Yeah.
So if the time is very short, we have to look at both cells, make sure that cancer is not developing or mutation hasn't formed.
That's very good. So I'm writing here protein/mutations, so that you could also take these cells from each group and analyze them and see, is there any mutations that this jelly juice is doing that would be something really bad if that would happen to a human. So that's really good. So time, proteins. So I guess the common denominator here is you're giving a specific thing that you're looking at. You're not just saying, we have 3 groups and we're going to assess regeneration, because then your judge is going to be like, well, how are you going to assess? And then you're going to say, well, I'm going to assess time. And time is an objective measure. It's not like a qualitative thing of saying, I'm just observing and seeing what's happening. So that's really important. You have to be very specific in terms of what you're measuring. And then you went a step further and said, I'm going to assess if there's any mutations or change in the protein levels and stuff like that. So that's really good. And then something George mentioned is, what would be your sample size? And people call that N number, N being the number of samples that you'll have in each group. So what do you guys think is a good sample size here?
Not all at once.
So it's not that easy, right? Can it be 1? Can it be 1,000? Can it be 2,000? What do you guys think?
What number feels right?
So should you go on a guess? Good. So you're saying you're doing research. So you would probably go look up people that have done a similar experiment and see how much they used. That would be a good measure, right? Instead of doing, you pull out and you say, I will do 10 of each group. And then your judge will be like, well, how did you come up with that number? So if you say, well, I looked up on this article and it said that they used this much, so you did the same, that is actually a very good, and that's what they do in a real research lab, right? They've never done it.
On your written report, did you mention why you're using that sample size? Like, I just saw in this article that they use
You don't need to necessarily. You could say that you based your experiment off one that's been done in another study. So that would be a better way of saying it in a more concise way. So that way, all the details of the experiment, all the N numbers and stuff will be inferred. Like, you don't need to explain one. You could say you got it from that study, but you still have to say in your proposal that you will use this many in each group. You will be measuring these specific variables. So that's important. But the reasoning behind it, you could just say that you're using it because another study has done it. And I think in the student guide, it actually tells you the exact things that you need to say in your experiment for you to get all your points. So did this exercise kind of help you guys to kind of visualize how this will work? It was very difficult to extrapolate from jelly juice, but
That was good.
I guess it's doable.
Thanks for Jamie.
Yeah. What else? You want us to check online if people have questions?
Yeah. Do you want to answer them live?
Yeah, we'll answer them live. So
This one right here.
so someone asked, are similar experiments allowed to be used to develop our experiment further?
Where's the mic?
It's on the table. They'll probably.
Yeah.
So are similar experiments allowed to be used to develop our experiment further? Not sure what they mean by that. You could use an experiment that's been done in another research study. So that's fine. But I'm not sure what they mean by experiment further, because typically you'll have to wait for the results of your study and then do the next experiment. But in your written proposals, you can include a section called future directions and say what you will do in the future. So for example, let's say Jilly Juice was faster at reproducing these cells. You will say in the future, we plan on doing this study on mice where we will be, I don't know, cutting their skin and giving them Jilly Juice and whatever. So that would be an example of what you would do to like a new experiment to complement yours. But you don't have to give the judge, the 5 or 6 different experiments that you will do. Focus on one, and then in your future direction, you could kind of extrapolate a little bit. But you don't need it. Your focus of your written proposal doesn't have to be all the experiments that you would do to test your hypothesis.
You can also answer that, like, if they mean it this way, maybe they're talking about using a similar technique in a different scenario, but they're trying to use the same technique. So let's say they're trying to test, I don't know, some sort of like drug delivery thing that was used successfully in treating cancer, right? Now I want to use the same drug delivery thing to treat, I don't know, like salmonella, right? If my research proposal is on salmonella, I want to use this drug delivery thing. The technique was used in another study. I can absolutely say that my research proposal that this technique was used for the cancer study. It worked for these reasons. This is why I think it will also be effective in my study on salmonella.
Yeah. So Maria, hopefully that answers your question. We'll go to OSA that says approximately how many teams are there usually in the final round? In the first round, I think we have 110 or 120, something like teams.
Okay.
Yeah. In the final round, I think we're anticipating 30. So I guess that's like a quarter chance. But honestly, a lot of people don't submit a proposal. Like they won't reach the final end goal. So if you guys actually just put in the effort and just give in a written proposal, that should get you like a lot of the way to get into the final round. So I suggest that just work on trying to get a final product for the teams to look at, even if it may not be fully baked by the time you're done.
Say there's 2 teams in school.
Yeah.
and you're just 1 team.
No.
Okay.
We don't do anything by school. It's whatever the top 30 teams are, they will progress. And we may take more than 30 if we get more money, but right now it's just 30 teams. Because we have to pay for like everything, like even your poster and everything at U Ottawa. So for now, it'll be just 30 and hopefully we could get more. But yeah. Yep.
Alright, so if there's specific scientific point of view, like, when I think about this, I feel like you guys would focus more on biology, chemistry. But given this and science, is that considered, or do you not consider that?
No, that is definitely real science. I wouldn't say it's not a real science. So, so one of the rules is you cannot do a clinical trial. Like you cannot design a study where it's like, I will use this number of patients and this patients and randomize them and stuff. That's very complicated, and that would not happen as the first step of your new hypothesis. Because the point of this competition is you're thinking of a new hypothesis, and usually when you come up with a new hypothesis, you're not jumping the gun and testing it on patients. You're going to study it on something like cells or whatever, right? Or plants or animals. But so social science, you can study, there are ways to study on the perspective of social sciences that would not be like human experimentation, right? But I can't really think, like, you mean like surveying and stuff like that, or like?
Yeah, but like, not everything about the experiment. Can't really have them without technology
Mhm.
on.
Right, yeah.
Yeah, also, these experiments, we're not actually doing them.
No,
Yeah.
no, you're just proposing.
I mean, if you want to look for a professor and do like actual research, that's another.
Yeah, I mean, I don't know if we had like, I think I've heard of one past participant where they tried to contact professors and propose like, I did this for the competition. Are you willing to do like, bring me in your lab and we actually do it? I don't know if they actually went through with it, but some people have gone through that length. I would say the purpose of the competition is to give you the opportunity to think like an actual researcher, right? Because if you're going to a lab, especially at your stage right now, you would be doing like pretty much the grunt work. Everyone starts as the grunt work. I mean, we've all been there. You know, you're doing basic tasks. The ideas you're going to be doing are not yours, right? They're whatever the person that's supervising you is giving to you. So this is going to be like one of the first opportunities where it's like you will be the one creating the experiment and creating this new idea. So I think that's the purpose. And the beauty of it is you just need a computer and your mind to do it. You don't need money and you don't need a lab to do that. So hopefully you'll get that out of the competition.
There's kind of a similar question, but I just wanted to go like, let's say you research about this chemical and you kind of want to base your procedure on that, your hypothesis on that, but it's already been kind of experimented with. Are you still allowed to use that, or do you have to be more innovative? Like, does it have to never been like a similar procedure has not been?
Yeah, so basically the medication or drug or chemical that you're referring to shouldn't be used for the purpose that you're testing for. So like, I guess the simplest way to explain what this creating a research proposal is in research, like every research group is very targeted to what they're looking at. So research group in Ottawa is very targeted at this chemicals will be used for cancer. And then another group and somewhere else in the world is going to publish stuff that you're going to find online on this chemical gets delivered in the body in this way and so on. But these research groups don't talk to each other often, right? So you looking in the literature will be reading about the chemical in different ways, and then you will be able to connect these different ideas together into this new research proposal. So that's the purpose of the exercise.
But it's like, it's discouraged to be like, they did this study and I'm gonna do the exact same
Right.
study
Yeah,
again.
you will lose points on novelty.
Yeah.
There is like, I think 30% of the points is just like, is this novel or are you just copying someone? So
So you can like incorporate somebody else's.
absolutely, yeah. And I think like the easiest way to explain that is like, let's say you're doing the topic is the opioid crisis, right?
Opioid crisis.
I guess the easiest way to explain this is like, let's say you have to pick something that you want to fix in the opioid crisis. So let's say, I'm not really sure what you would like. Someone give me an example of what they
Genetic susceptibility.
genetic susceptibility. Perfect. So let's say you're hypothesizing. Okay, let's say you research that gene
ABC.
OP for
Okay.
opioid. Okay, you're hypothesizing that gene OP will make someone very susceptible to drug overdose or like addiction. Addiction. So addiction. Okay. So let's say that's what you're hypothesizing. So you're thinking, okay, what's going wrong with gene OP? So then you're researching and you're saying, oh, gene OP, there is, let's say it makes some sort of protein, and when it's very low levels in your body, that means you're going to get addiction. So then you're like, okay, I'm going to try to find something that will increase these levels of this enzyme or protein or whatever, right? Because when it's low, that's what's giving me an addiction. So then you will research, well, how can I increase this? And then you discover drug X, and some researcher discovered that drug X will increase the protein. So then you will put everything this together and you'll say, drug X, I will give that, it will increase this protein. And because I know this protein will, when it's low, it causes addiction. Right? If I increase it, that will stop the addiction. So basically it's like, you want to get to Z, you know Y is there, Y goes to Z, and then you know X goes to Z, therefore X goes to Y, therefore X will get me to Z. Is that kind of clear? I don't know. Yeah, it is? Okay, yeah. I realized it was a little bit confusing, but yeah, that's basically what you guys are doing. And if you're able to do this exercise well, you will find that you will find a very good proposal. Yeah.
Okay, we have a question from the chat. The question asks, suggestions from blogs or websites or other sources to generate an idea. So I guess for that question, they're asking, what can we use, what sources can we use in order to generate an idea for the proposal? What I would suggest is that you look at previous literature, that being on Google Scholar, as you mentioned. There's also PubMed. You want to look at previous literature that kind of targets the field that you want to, or the approach that you want to take. I don't know if you guys want to add on to
I
that.
can add on to that, yeah.
Yeah,
So essentially, when you're looking at like blogs and websites, it's a great place to start, but you don't want to make your whole research proposal on the basis of that blog. You can't be like, on this blog, Jenny said that like you can, or Joey said you can regrow arms because you drink juice, right? Like it's not backed by science. You can start there and give you some ideas of where you want to go with your research, but like Justin said, you want to back up the research in your actual research proposal with academic sources. So whether that be from Scholar, from PubMed, you want to make sure that you have concrete evidence in support of theory and of techniques to actually go through with your research proposal. But absolutely start with some blogs, start with some like Wikipedia even. These are good places to start to give you ideas and some general understandings. Often the information is more condensed and easier to read and maybe even more enjoyable to read. So good place to start, but not great place to stick with for the actual support of your research.
Are there any more questions?
questions, yeah. In the chat below, there's no questions.
We'll give them one last shot.
Do you guys have any questions? Yeah, go for it.
Sorry, one more thing. Can we access the slideshows?
Yeah, we'll send them out.
We'll wait 30 more seconds and then we'll end it. I'm sure some of you will ask us questions when all this is over. So we'll just wait like 30 more seconds for anyone at home to ask their questions, and then we'll end the live stream. The YouTube link is going to be shared to everyone. Oh yeah, did you have a question? Oh, you were just on mute. The YouTube link is going to be, it's already shared to your emails. You might have gotten it already. So you'll have a recording of this session that you can review, and then you'll have the PowerPoint sent to you. So you'll have all of that. The YouTube stream started like a little late, obviously. So you won't have like the beginning, but George did a little summary during the break. So hopefully that helps. Okay, so that's the session. I'll be ending the live stream. All right, no problem. So let me just just end it here.